Proteins aren't just blobs of amino acids. They have structure, they have form, they have domains.
I'm just a simple chain of amino acids. No folds, no drama, but I hold all the instructions.
I start the folding. I form alpha helices and beta sheets. I will bring some shape into this protein party.
Look at me now, all folded and ready for action. I've got bonds holding me tight. Hydrogen, ionic, disulfide, you name it. And of course, me. I bring multiple chains together. Team effort.
You may look good, but I do the job. Catalysis, I'm on it. DNA binding, that's me. Signaling, yup, I'm the domain, the working unit of the protein.
So it means that without you, we're just structure.
So you're like the hands and tools of the protein?
Exactly. One protein, many domains, each with a special task.
In this presentation, let's break down what protein domains really are, how they help protein work, and where we find them in real life.
Today, we'll be tackling about domains in protein structure, functional units. For our learning outcomes, at the end of the lesson, the students are expected to first define what a protein domain is, identify different types of domains and their functions, explain how domains help proteins perform specific tasks, and lastly, give examples of domains found in real proteins and their application and relevance.
Okay, so let's start with visualizing protein. Let's think of it as a building block. Each level of structure adds complexity and shape to its overall design and function. Let's first talk about primary structure, which is the blueprint. This is simply the sequence of amino acids in a polypeptide chain. Let's think of it as an alphabet or sentences of a protein, and even one small change of this can affect the entire protein's behavior. For example, methionine, glycine, serine, and leucine. Next is the secondary structure where the folding begins. Now the chain begins to fold locally. Common shapes like alpha helix, which is spiral, and beta pleated sheet, which is zigzag. It's stabilized by hydrogen bonds between backbone atoms. Next is the tertiary structure, which is the 3D shape emerges. This is the overall 3D shape of the entire polypeptide. It involves interaction between R groups like the side chains, hydrogen bonds, ionic bonds, hydrophobic interactions, disulfide bridges, and this is where the proteins becomes functionally active. Like for example, an enzyme to fold to create its active site. Then next is the quaternary structure, which refers to how two or more separate polypeptide chains, which is called the subunits, come together and arrange themselves into a single functional protein complex. Now, why does 3D structure matter? The 3D structure of a protein determines its specific function, whether it's binding to another molecule, catalyzing reaction, or giving support to a cell. Like for example, shape is equal to function. So if a protein is misfolded, it doesn't work properly. And enzymes, for example, have an active site that matches their substrates like a lock and key. And also, changes in pH temperature or mutations can destroy the structure, which can cause the protein to denature and lose its function.
Now, let's check our understanding of protein structure. Which of the following best describes the tertiary structure of a protein? If your answer is C, that is correct. It's the overall 3D shape formed by R group interactions.
Now let's talk about the protein domains. Think of a protein like a big house, and inside that house are different rooms. These rooms are the domains. Each room has a specific purpose. Maybe one is a kitchen, another a bedroom, and another a study. Just like that, each domain in a protein does a unique job. One might bind to DNA, another might catalyze a reaction, and another might be help the protein interact with other molecules. Now there are two main kinds of domains, structural domains and functional domains. Now structural domains are like the architecture of the room, the walls, the ceiling, and the furniture. They fold into a specific stable shape, and even if you take them out of the protein, they still hold their form. On the other hand, functional domains are the parts that actually do something, like for example, a stove in the kitchen or a desk in the study. They carry out the protein's main task. It's from the word itself, functional. Now, here's where it gets cool. Domains are modular, meaning they're like Lego blocks. You can reuse them in different proteins. Evolution has been doing this for ages, combining these domains to make new proteins with different jobs. And most domains can even fold on their own, like a pop-up tent. They don't need help from the rest of the proteins to take their shape. That makes them super versatile. So in short, protein domains are like specialized rooms of Lego pieces, each with a structure, a function, and the ability to work independently and be used in different biological houses.
Now, let's have another checkpoint. Why are protein domains considered modular? That's right. The answer is letter C. It's reused and functional in different proteins.
Let's talk about the functional roles of domains. First is catalytic domains. These are the parts of a protein that perform chemical reactions usually found in enzymes. In these domains, amino acids are arranged in such a way that they can interact with a substrate, break or form a chemical bonds, and convert molecules from one form to another. One of the great examples here is the kinase domain, which transfers phosphates group from ATP to a target molecule. Catalytic domains are like the hands of a protein that do the actual work in a reaction. Next is the binding domains. These domains help the protein recognize and attach to the molecules such as the RNA, DNA, small ligands like hormones or ions. There are several types depending on what the domain binds to, like the DNA binding domains, which is zinc fingers, helix turn, helix motifs are commonly found in transcription factors and help control gene expression. Then for RNA binding domains, which are found in proteins that regulate splicing, translation, or RNA stability. And then for the ligand binding domains, bind to small molecules like hormones, which often trigger a change in a protein shape or activity. Basically, this domain, think of this as a lock. They are specially shaped to fit a certain key, whether it's a DNA segment, a molecule, or a signal. Next is the transmembrane domains, which are found in proteins that span cell membrane. These domains are made up of hydrophobic, which is a water-repelling amino acids that allow the protein to pass through or anchor into the lipid layer of membranes. These are very common in receptors, ion channels, and transport proteins. For example, the insulin receptor has transmembrane domains that anchor it in the cell membrane, allowing it to sense insulin outside the cell and send a signal inside. And lastly is the protein-protein interaction domain. These domains allow proteins to physically attach to another to form complexes or pass signal along a pathway. Some well-known examples here is the SH2 and SH3 domains in signaling proteins, PDZ domains in scaffolding proteins, leucine zipper motifs, and transcription factors. So these domains are essential in cell communication, signal transduction, and forming multiple complexes, much like Velcro strips that allows proteins to snap together and work.
Let's have another checkpoint. Which domain is responsible for breaking or forming bonds in chemical reactions? The answer is letter B. It's catalytic domains. Next question. That's right. The answer is C. It's the binding domains.
Moving on, we have the domain examples in proteins. So I will be giving different analogies with the different examples for us to be able to understand them better. So first we have zinc fingers. So these are the protein structures that bind to DNA or RNA and are crucial for regulating gene expression and other cellular processes. So they're like a hand holding on to a rope. So they grab and hold on to DNA to control what happens in the cell. Next, we have the kinase domains. So these are protein domains that act as enzymes. So they catalyze the transfer of a phosphate group from ATP to other molecules, which is a process called phosphorylation. So they're like putting a sticker on something to mark it. So they add a phosphate sticker to tell them what to do. Next, we have the SH2 and SH3. So these are protein modules that mediate protein-protein interaction. So they play a crucial role in signal transduction pathways, particularly those initiated by tyrosine kinases. SH2 domains specifically bind to phosphotyrosine containing sequences in target proteins, while SH3 domains recognize protein-rich motifs. So for our analogy, these are like friends holding hands. So they help proteins find and stick to the right person. And lastly, we have the immunoglobulin domains. So these are found in proteins like antibodies. So they play a crucial role in the immune system. So they're like a shield that can grab bad guys. So they help the immune system catch germs and protect the body.
Let's check our understanding on some domain examples in proteins. True or false? SH2 and SH3 domains break down proteins during digestion. If your answer is false, you are correct. They are for protein-protein interaction.
Protein domains are not just structural features, so they also have real-world applications. So in bioinformatics, identifying known domains in a new protein helps us predict its function, location, and interactions even without doing lab experiments. So this speeds up protein annotation and advances our understanding of how cells work. Next, in protein engineering, domains are like building blocks. So since they can function independently, scientists can mix and match them to design custom proteins with new or improved functions, like tools with added features. And finally, in medicine, many diseases are caused by mutations in functional domains. So these changes can disrupt a protein's job or even make it harmful. Knowing which domain is affected helps researchers also to develop targeted treatments and diagnostics, making domain knowledge crucial in personalized medicine. So whether in research, biotech, or healthcare, protein domains play a central role.
Now, let's try to answer this final question. How are protein domains used in protein engineering? The correct answer is letter B, to mix and match functional parts to create new or improved proteins.
So, to summarize everything on the domains in proteins and its functional units, we have the levels of protein structure. So first, we have the primary structure. So this is simply the amino acid sequence, and even a single change here can affect the entire protein's function. Next is the secondary structure. So this involves local folding into shapes like alpha helices and beta sheets, and they are held together by hydrogen bonds. Next is the tertiary structure. So this is the full 3D shape formed by the interactions between side chains. So this is where the protein becomes functional. And finally, we have the quaternary structure. So this happens when two or more chains come together to form a complete working protein. Next, we have domain. So a domain is a part of a protein that folds independently and does a specific job. So think of it like a tool or a room in a protein house. Now, for the functional roles of domains, we have four. We have the catalytic domains, binding domains, transmembrane domains, and interaction domains. For catalytic domains, they do chemical reactions. For binding domains, they attach to DNA, RNA, or small molecules. For transmembrane domains, they anchor protein in membranes. And lastly, for the interaction domains, they connect proteins in signaling and complexes. So, for the domain examples, we will give different analogies with the different domain examples. So for zinc finger, so they grab DNA, so they are like a hand holding on to a rope. For kinase, they add phosphate, so they are like putting a sticker to tell them what to do. For SH2 and SH3, they bind partners, so they are like friends holding hands. And lastly, for the immunoglobulin, they catch pathogens, so they are like a shield grabbing the bad guys. So all these are modular, so they are reused across many proteins. Now, for the applications and relevance, so we have the bioinformatics. So domains help predict unknown protein functions. So the keyword is predict. For protein engineering, they mix match domains to create new proteins. So the keyword is mix match. Lastly, for the medical use, so mutations in domains cause diseases. Targeting them helps in treatment. So the keyword is treatment.